Cadherins, derived from calcium-dependent adhesion, serve as vital cell adhesion molecules necessary for establishing adherens junctions that enable cells to stick together. They belong to the category of type 1 transmembrane proteins reliant on calcium ions for their functioning. Facilitating cell-cell adhesion are the extracellular cadherin domains, while the intracellular cytoplasmic tail connects with multiple adapters and signaling proteins collectively known as the cadherin. The significance of the cadherin family lies in its role in sustaining cell-cell contact and overseeing cytoskeletal complexes. This family encompasses various members such as cadherins, protocadherins, desmogleins, and desmocollins, among others. These proteins share cadherin repeats within their structure, serving as the extracellular Ca2+ binding domains. Diverse classes of cadherin molecules are assigned prefixes based on the tissues they associate with classical cadherins forming homodimers in cis to maintain tissue integrity, while desmosomal cadherins exist as heterodimers, focusing on offering resistance to cellular damage. Atypical cadherins like CELSR-AH1 maintain extracellular repeats binding activities of other cadherins, but differ structurally, primarily involved in relaying developmental signals rather than adhesion. The specificity of cell clustering based on cadherin subtypes indicates their critical role in cellular organization, evident in cell culture experiments, as well as during development. The differentiation between cadherin subtypes appears to be based on kinetic specificity, reflecting varying lifetimes of different cadherin homotypic bonds. Cadherins undergo extensive post-translational modifications to facilitate cell-cell adhesion and recognition, beginning as polypeptides approximately 720-750 amino acids long. The transmembrane component consists of single-chain glycoprotein repeats, housing 5 extracellular domain repeats that serve as Ca2+ ion binding sites, critical for cadherin function. Over 100 types of cadherins have been identified and sequenced in humans to date. Homodimeric cadherins play a pivotal role in forming cell-cell adhesion by transitioning from cis-dimers to transdimers when interacting with cadherins on neighboring cell membranes, culminating in the establishment of adherens junctions facilitated by protein complexes like A, V, and G catenins binding to the cadherin's cytoplasmic portion. The regulatory proteins linked with cadherins include p-Chapern-20 catenin, A-catenin, V-catenin, and vinculin, each contributing to enhancing classical cadherin stability through varied interactions. Cadherins' functionality extends to their involvement in cellular movement and tissue structure maintenance post-development, with regulatory mechanisms like promoter methylation modulating cadherin expression levels. The disruption of N-cadherins can impede cardiomyocyte development, leading to cardiac anomalies and functional impairments. Cadherins also actively participate in cell migration events like the epithelial mesenchymal transition in early development, critical for the formation of various tissues and organs. Furthermore, cadherins implicated in the epithelial mesenchymal transition have shown relevance in the reprogramming of adult cells into induced pluripotent stem cells, expanding their significance beyond developmental processes. The association of cadherins, particularly E-cadherins, with tumor metastasis underscores their role in modulating cellular adhesion and motility, where disruptions in E-cadherin-mediated adhesion are linked to increased tumor invasiveness and metastatic potential. Cadherins' involvement in cancer progression extends to various forms of carcinomas, where alterations in cadherin expression levels can lead to abnormal cell signaling and uncontrolled cancer cell proliferation, potentially fostering the development of secondary malignancies. Additionally, advancements in research aim to decipher the impact of cadherins on tumor growth and metastasis, emphasizing their potential therapeutic implications.